And welcome to the discussion on the process of cell division. As I said, I'll be showing you two short little videos. The first one here will deal with chromosomes in the cell cycle, and the second one we will get into mitosis and cell division. So let's get going. Okay, so the first thing I need to discuss before we get into cell division is chromosomes. And chromosomes, what are chromosomes? Well, first of all, chromosomes are tightly wound DNA. Okay, chromosomes only appear during cell division. Okay, and once that cell division is completed, they will unwind themselves into what is called chromatin, which is loosely bound DNA. Okay, what's the importance of chromosomes? Since chromosomes are made up in DNA, they carry the genetic information that gets passed from one cell to another or from one generation to the next. The reason that the DNA winds up or binds itself into chromosomes is this makes it much easier and to separate the DNA during cell division. It also makes sure that the DNA can be copied more precisely and passed on from one generation to the next. This DNA must copy information before cell division begins. It does that through a process called replication. Again, forming chromosomes allows us to make this easier, which then allows each new daughter cell to get its own copy of the genetic information in full detail. One thing you need to understand is that each type of organism has its own specific number of chromosomes. For example, in humans, our chromosome number is 46, and we call that the diploid number. So some things about chromosomes and the importance of DNA winding up into chromosomes. Okay. Now, I need to talk about differences between prokaryotic and eukaryotic. There are differences in the chromosomes between the two. Again, prokaryotic are your single-celled simple organisms with no nuclei. Eukaryotics are the ones that have nuclei and much more complex chromosomes. And you can see here in the picture, again, prokaryotic cells lack nuclei. Their DNA is found simply in the cytoplasm. Because of this, there is simply a single circular DNA strand. Okay, it is just one single circular DNA strand. This is much different than what we have in eukaryotic chromosomes. Okay, eukaryotic chromosomes, the DNA is contained, as we said, in the nucleus, and it is made up of chromatin. This is loosely coiled DNA in histone. When it comes time for cell division, this chromatin will start binding itself up to form chromosomes, and instead of having one circular strand of DNA, we have multiple chromosomes depending again on the type of organism. Those chromosomes have a tendency to pair themselves up. Okay, and this is what helps with cell division, or what helps with the splitting of the genetic material during cell division. Okay. This coiling up of the DNA around the histone creates something called a nucleosome. These nucleosomes then interact with one another to form the coil and supercoils that make up chromosomes. Okay, so again, chromosomes in eukaryotic organisms is much more complex than what we find in prokaryotic organisms. So, with that said, now we can get into what is called the cell cycle, the life of a cell. Okay, again, we're going to distinguish between prokaryotes and eukaryotes. Let's do prokaryotes first. It's much simpler. A prokaryotic organism will simply go through a Retter pattern of growth, DNA replication, cell division. The cell will replicate or copy its DNA once that cell reaches a certain size. Again, remember we talked about cells like to be small. When they reach a certain size, they become less efficient. The DNA is stressed, so they have to divide for better efficiency. When the DNA replication is complete, the cell will simply undergo a process known as binary fission. In other words, once the DNA is duplicated, okay, once that strand or that circular strand is duplicated and you have two circular strands, the cell will simply split in half, with each circular strand going into a different cell, and you have two identical prokaryotic organisms. Okay, again, prokaryotes, cell division is much simpler than what we have with eukaryotes. Okay. Eukaryotes, as I just mentioned, much more complex. It is made up of 4 different phases, G1, S, G2, and then the M phase. These cells must go through all of these processes to make sure that the DNA and cell are prepared for cell division so that each of the new daughter cells has all the stuff that it's going to need to function properly. So as I said, there are 4 phases, the G1, GAP1, S for synthesis, G2, GAP2, and M, or mitotic phase. Okay, and we'll be talking about these. Now, the first thing I need to talk about is how a cell spends most of its life. It spends most of its life in a phase called interphase. This consists of the G1, S, and G2 phases of the cell cycle. Basically, interphase is the time between cell divisions. It is a period of growth for the cell. It is also when it carries out all its life activities and carries out all the functions that it is designed for. Okay, the M phase is cell division. Okay, this is where the cell will go through a process of dividing up the genetic material and producing 2 identical daughter cells. Okay, so we have interphase and the M phase. The interphase consists of the G1, S, and G2. Cell division itself actually is just made up of the M phase, which consists of mitosis and cytokinesis. So let's talk here about GAP1 phase. This is mainly cell growth. It increases in size. It synthesizes new proteins. It synthesizes new organelles because each new cell that's going to get produced needs its own proteins and organelles. So once those new proteins and organelles are synthesized or made, they can be split up when the cell divides. So again, this is just a period of cell growth necessary so that each new cell will have all the necessary material that it has. From there, we go into the S phase. The S stands for synthesis. What we are synthesizing is DNA. We are making a copy of the chromosomes. Okay, this process is called replication. Okay, this process is called replication. When this happens, we end up with chromosomes consisting of 2 chromatids. You can see here that chromosomes, the paired chromosomes, and I told you that chromosomes like to occur in pairs. They duplicate themselves. Each new strand is attached together at a center point called a centromere. We'll talk about that more. So each new chromosome is double-stranded. So we go from single-stranded chromosomes to double-stranded chromosomes. Again, they are in pairs. From there, we go into a preparation for cell division. Special organelles, especially if you're talking about organelles in animal cells, have to be created. Those organelles are called centrioles, and they occur in pairs also, and they are necessary for cell division. Okay, some other structures that go along with the centrioles, such as spindle fibers, begin to form, and all of this is necessary for the separation of the genetic material. Okay, so we have all of those things taking place. Okay, another thing that happens here is that errors or any errors from replication are fixed. Okay, when that DNA copies itself, sometimes there are errors that occur. There is a checkpoint here that the cell must go through during G2, and if it locates any errors, it will fix them. If it cannot fix the errors, then the cell will go through a process called apoptosis that will actually, the cell will die off and not be reproduced or copied, and then we move on. Okay, those errors are not good. Now, some errors do get through, don't get me wrong, that's what we call mutations. Okay, but we try to eliminate, or at least the cells try to eliminate, any errors that have occurred. From there, we will enter the M phase, the actual cell division of the cell. Okay, this occurs in two parts, as I mentioned. Okay, there is mitosis, which is actually the division of the cell chromatin, the genetic material, the DNA, however you want to say it. And then there is a process called cytokinesis, and this is where the cell actually divides. The cell membrane pinches in, the cytoplasm is divided in half, with half of the chromatin or half the genetic material going into each new cell. So, this is where I'm going to stop on this first little video. That is the cell cycle. Okay, in the next part, I'm going to talk about actual mitosis and cytokinesis. Okay, so just move on to that next part and go from there.